Adhikari here. So, as usual, let me introduce a little bit our speaker before he starts. So, he obtained his bachelor degree at USP, so nearby, in chemistry. And then he moved straight to US, and he got a PhD in neuroscience from Columbia University in 2011. And then, as usual, he had a postdoc career that was concluded in 2017 with a postdoctor in Stanford University. And after that, he moved to UCLA, where he started his own lab, where he's working right now. So his lab studies, in general, brain activities and brain circuits that control fear and eating in mice. And this work has been published by major journals like Nature and Nature Communications and so on. And his lab discovered brain circuits, that increase and suppress the anxiety and fear, so this kind of, let's say, feelings. And today he's gonna talk about how we can use visible light and viruses to study brain functions. So the floor is yours.
Okay, thank you, Dario, for the invitation and for the generous introduction. So today I'll talk a little about the history of the studying of the brain from antiquity to modern age, and then how more recently we have developed new methods to answer questions that people were interested in exploring but couldn't do until recently. So first, this quick introduction about me. So I'm from Brazil, and then I did my bachelor's here in Sao Paulo, then I did my PhD in New York, and since then I've been in California after doing my postdoc also there, studying the neural circuits that control feeding and fear-related behaviors. So first, since this is not my usual audience of other neuroscientists, I thought it would be good to start with a motivation of, like, what is neuroscience, first of all? So neuroscience is a part of biology that studies the brain and behavior and mental function and why we get hungry or sleep and have diseases of the brain. But it's not a part of medicine. It's a part of biology that studies the brain. And then in contrast, neurology is a part of medicine that studies and treats patients that have issues like strokes and epilepsy. And psychiatry is also not a part of biology. It's also a part of medicine that studies other mental issues like schizophrenia or anxiety disorders. So that's first just a quick definition of what is neuroscience. And then why should we study neuroscience and the brain? And there are many reasons to do so. So here I highlighted 3 of them. So the first is to understand who and what we are, because that's a question that people have been pondering about for many millennia, since when there were people, like why we act the way we do and what makes us be the way we are. And we can also use knowledge from neuroscience to improve the life quality of patients that are treated by the neurologists and psychiatrists to develop better therapies. And lastly, more recently, you probably have heard of these many interesting developments in artificial intelligence that attempts to reverse engineer some of the processes that happen in the brain to then develop new artificial machines and systems that can solve lots of problems that the brain can solve. So these are 3 among many of the other reasons to study the brain. Oh, and also if you have any questions, please raise your hand and interrupt during the talk. Okay. So first, let's start from the very beginning of neuroscience. So people are always interested in each other's behaviors and emotions and memories and things like that. So that's not a new type of curiosity that always existed in people. But in antiquity, people didn't really think that the brain was important for those processes. And instead, they thought that the heart was the center of the emotions and a lot of mental activity. And that kind of does make some intuitive sense, because when you are agitated or calm, you don't feel anything different in your head where the brain is. But instead you feel that maybe your heart accelerates and you feel heart palpitations when you're nervous. So people really thought that the heart was the center of emotional life. And then that's why we have these words now like kind-hearted or generous heart or cold-hearted is because the heart was thought to be the center of emotions. But in reality, it should be something like, that person has such a generous brain, they're so nice. But that's how it started. And because the brain was not viewed as important, the pharaohs kind of preserved a lot of their other organs, but they threw out their brains. So they never preserved their brains after the pharaohs were dead. Okay, and then kind of intellectuals from antiquity, like Aristotle, studied the brain, and then he observed that the brain had many folds in it, so it's not a smooth surface, but it's a very folded surface. So Aristotle thought that the brain's function was to release and dissipate body heat. So you get very hot, and then the heat goes up to the brain and is dissipated. So he thought that's the function of the brain. And the first kind of insight that really made people think that the brain had something to do with behaviors and mental function came from the ancient physician Galen, who also treated some of these gladiators. So the gladiators are these strong people that fought each other for other crowds' entertainments, and then sometimes they had head damage from concussions and blows and attacks and things like that. And then Galen was the first to observe that sometimes before the fight, the gladiator was talking okay and moving correctly, and then after the fight, after they had some kind of injury to the head, sometimes they lost some kind of movement function, or their personality might change a little, or they were unable to move or feel certain things from other parts of the body. So this was the first time that people thought there was some evidence that the brain was controlling the behavior and not the heart. Okay. And after people thought that the brain was important for controlling mental function, that really increased the attention that the scientific community paid to the brain. And the early anatomists like Leonardo da Vinci also paid a lot of attention to the brain. And that's where we get some of our first anatomical drawings of what the brain looks like. So when he was dissecting bodies and drawing what the brain looked like, these anatomists observed that the brain had these kind of holes in them. And these holes were filled with a fluid, so some kind of liquid. So they observed that. And then they also saw that there are these nerves that come out of the brain. And the nerves go from the brain to other body parts, like the hand and the leg. So they inferred that, oh, perhaps the brain is using these nerves that goes from the brain to the leg to perhaps move the leg or feel sensations from the leg, which turned out to be a correct insight. But in the history of neuroscience, whenever there's a correct insight, there's also some wrong idea that often goes along with it. So even though they correctly thought that the movement of the limbs was caused by the nerves, they also thought that these fluid-filled chambers made the limbs move hydraulically. So they thought that the fluid in these holes of the brain went inside the nerves, and they kind of hydraulically made the movements that people are making. So they had this wrong idea. So there was one advanced, but one kind of wrong idea. And after that, there was another important insight, this time really closely linked to physics. So in the 1700s, the physicists were very interested in studying electricity, and they studied a lot of atmospheric electricity and static electricity and things like that. And here, Luigi Galvani, who was also very important in physics, discovered for the first time the link between electricity and muscle movement and muscle contraction. So he did this famous experiment in which he got legs from recently killed frogs. And then he discovered that if you pass electrical current through the nerves of the legs of the legs kind of moved as if they were living. So that really was a very cool finding that showed people that electricity that was being studied by physicists could also be related to causing movements in animals. And because it looks like the frog legs kind of became living again, they did lots of demonstrations with this phenomena. So sometimes after they decapitated like a prisoner, They got their freshly cut head and then put electricity through the nerves, and then the prisoner's head made all sorts of movements and then sounds like this. So they really tried to use electricity to reanimate dead bodies. And this was something that inspired one of the most famous books, which is Frankenstein, which was written slightly after this time, but it kind of used these scientific discoveries that are happening at that time. Okay, so at this point, people know that electricity is important for movement and that the brain is important for mental function, but it wasn't known if different parts of the brain have the same or different functions. So perhaps all of the brain has the same function. We don't know. Then one important discovery that showed that different parts of the brain might have different functions came from this very famous case study here. So this is a guy called Phineas Gage, who was a very hard worker person who worked in the railways. So he worked in putting on new railways tracks, which is a very dangerous, laborious job. And then he was very good at that activity. And one day there was an explosion, and a very large piece of metal was propelled through his head like this. So this big metal rod kind of went through his head. And then, miraculously, he didn't die. So he recovered from the injury, but now he had a lot of brain injury because that thing went through his head. And after the injury, his personality changed a lot. So before he was very mild-mannered and humble, and then after the accident, he became very impulsive and aggressive and violent. So he had this change in some personality traits, but some other mental functions were still working properly. So he was, he wasn't blind, he was able to see properly, he was able to move all his hands and feet correctly, he could walk. So he seemed to have brain damage in a particular area, and that caused problems in one type of mental function, which was kind of impulsive control. So that suggested to people that different parts of the brain might have different functions for the first time. And then following up on this insight, people started studying the brain more rigorously in laboratory settings, and also found that indeed in the lab, if you do more careful experiments, you can see that different parts of the brain have different functions. And some of these early experiments were by making controlled lesions in different parts of the brain. So you make a lesion either in this nucleus of the brain or in that other region, and then you could see that sometimes different things happen to the animal. And this led to one famous finding, which is that there's a region in the brain called the lateral hypothalamus and another called the ventromedial hypothalamus. And then if you lesion the ventromedial hypothalamus part of the brain, they observed that rats became very obese. So it's a normal athletic-looking rat, and then if you lesion this particular nucleus, rats become very fat, which suggests that the normal function of this brain region is to decrease eating. And then they also found another brain region that if you lesion, it makes rats become very thin, suggesting that the function of that region was to make the rat eat more. So lots of studies like this suggested again that this initial view was correct, and then that different parts of the brain kind of have different functions. So at this point, we already know much more about the brain than the Pharaoh antiquity times, right? They know that the brain is important for mental function, that electricity is an important part of brain action, and then they also knew that different parts of the brain had different functions. And more recently, we have found out also that not only do different parts of the brain have different functions, but also that different brain cells in the same region might have different functions. Okay, so remember I said previously that lesions of the hypothalamus kind of could make the rat become fatter or thinner. And recently it was found that within the hypothalamus part of the brain, there are kind of these red cells and these blue cells which have opposite functions. So within a very tiny small part of the brain, there are these red cells which kind of increase hunger, and then there are these blue cells that decrease hunger. So has exactly the opposite role in eating. And those two types of cells are just next to each other. So they're in the same part of the brain, but opposite functions. And similarly, there's this other part of the brain called the striatum, and it has these two different types of cells that we call D1 and D2 cells. And activation of D1 cells facilitates movements and running and these sorts of things, while activation of the D2 cells suppresses movements and creates more immobility. So again, these are just two of many examples where you can see that even within one tiny region of the brain, there are often cells that have completely different or even opposite functions. And because of that, if you were to, say, lesion this whole brain region, then you would lesion and destroy both the red cell and the blue cell, and then it would be difficult to find out that these different cell types have different functions. So to study the function of unique individual cell types in the brain, we really need some kind of method that allows you to activate separately just one cell type without destroying the whole region of the brain. And
so the previous results that were, let's say, very rough, in which you just destroy something, let's say, they still create a macroscopic effect because these type of cells are unbalanced in a single region, I guess, right? So you destroy more, because you might guess, well, I destroy both the POMC and the AGRP.
Yeah.
They compensate, so nothing happened to a certain extent.
So
but they are unbalanced in a specific region, so you destroy more of one type. And
it can be many different options. You could get this idea that if I destroy this region and both have different functions, then nothing happens, so you get kind of a false negative result. That could happen. But it could also be that maybe, like you said, there's more of one cell type than the other, and then you do get some result, but there's some other different underlying result that it's not accessible to you. So this would be something like if you're trying to study how a car works, then if you destroy all the pedals, we'll correctly infer that the pedals have something to do with the car moving. But you'd have to separately activate the brake and the acceleration pedal to figure out that, oh, those two things are both pedals that are both near each other, but they have completely opposite functions in car movement. So just like that, it's the same kind of thinking, but in the brain. Yeah, any other questions? Yeah.
There is any rule of the proportionality between those cells in between species? So if you change the species, you have different ratio of proportionality between the cells?
Yeah, you can. Different species might have more of one type of neuron than the other, or may not.
Like a number? Do you have for example, that in the ratio between in the picture the blue cell, the blue neurons and the red ones? What it will be like the ratio between the both?
In this case, there's more of the blue one, but the more is not necessarily the important factor, because you can also have cells in the brain where there are very few of them, but they have a disproportionately large influence.
Okay.
And there's also times when there's more of one type of cell, but they have each one has very tiny influences.
Okay. So
because they maybe send electrical connections to many, many, many cells, even though there's few of them. So these red cells, there's very few of them in the brain, but they are very influential because they electrically connect to many different brain regions and then can produce lots of behaviors that bias the animal towards eating more food.
Okay, see.
So for example, one function that these red cells have is they cause more eating and hunger, but if the animal is eating more, it has to do less of some other things. So it can't be, for example, fighting and eating. So these cells not only promote hunger, but they also suppress circuits that promote fighting, because only one behavior can be happening. So they're very influential, even though there's few of them. Okay, but it seemed pretty clear that there's different cells with different functions, so then we had to discover some method that allows us to selectively activate just one type of neuron. And then this is where this optogenetics method came to solve. So before getting into the method, we just have to talk very briefly about how the neurons in the brain normally work and how they use electrical currents. So let's say we have these two neurons here, this brown one and this green one, and then they communicate at this location called the synapse. And what happens is the brown neuron will release a chemical molecule of some sort, and some of those molecules are neurotransmitters you have already heard of, like dopamine or serotonin. So the brown neuron will release a neurotransmitter molecule of some sort, and then the green neuron, if you zoom in this synapse here, see the brown releases some neurotransmitter molecule, which is this blue circle. Then the green neuron has some kind of protein that binds that neurotransmitter receptor, which could be, again, serotonin or dopamine or other ones. And after binding that neurotransmitter, eventually what happens is that some kind of electrical ion charge will go from the outside to the inside of the cell, or from the inside of the cell to the outside of the cell. So to simplify here, the most common type of synapse causes this to happen, which is after binding a neurotransmitter, it opens a little pore in the membrane, and then some electrically charged sodium ions go inside the green cell. So basically, in a very simplified manner, if enough positive sodium charges go inside the green cell, then the green cell becomes electrically active and creates an electrical impulse. And that's kind of how the neurons communicate with each other normally. It's one neuron releases a neurotransmitter, the other neuron has some protein that binds it, and then after binding it, some kind of ionic current change happens in the neuron. So what this shows is that if we were able to alter the flux of ions through the green neuron, then it would be possible to selectively control just the green neuron. Okay, and the way this happened, that we can do it now in modern neuroscience, is through this method called optogenetics. And this has a very interesting, strange history. So this method starts with some people that are not neuroscientists, and they're microbiologists, and they were studying unicellular algae. And the algae are this kind of green little unicellular algae that they absorb sunlight, and then they make their energy from the sunlight. So the algae needs to have some mechanism to detect when there is sunlight. So this unicellular algae had a protein called channel rhodopsin, which is shown here in purple. And this is the inside, this is the outside of the cell. Then in the presence of blue light, this channel rhodopsin protein absorbs the blue light, and then it opens a little pore in the algae, and then some positive sodium charge goes inside the algae because there are lots of ions outside in the water. And then the algae uses this for its own metabolic purposes. But this is very conceptually similar to how the neurons work in the animal brain, right? So in the animal brain, there is some kind of protein that binds a neurotransmitter and then lets sodium ions inside the nervous system cell. And here we have the algae absorbs not neurotransmitter, but blue light. And then the blue light opens a little channel that lets sodium go through the algae. So this seems like a way to potentially use this visible light to control the flux of ions inside a neuron. So someone had this idea that if we made the neurons produce this protein from the algae, then the neuron in the brain would become sensitive to blue light. Then it would absorb blue light and become electrically activated. And here we have a kind of diagram of what's happening. This protein absorbs blue light, and it opens a passage for sodium ions when the blue light is turned on, and then some sodium currents are seen that alter the voltage of this cell. Okay, so any questions about this part? Yeah. Okay, so this is how people thought it would be possible to use this naturally occurring protein from the algae to study brain function in neuroscience. So first, they figured out what would be the DNA sequence to make that protein from the algae. So that's step one, make a DNA sequence that has the information to make that channel rhodopsin protein. Then you put that DNA sequence inside a virus, because the virus normally has virus DNA, which is not useful for neuroscience. So we make a different virus that the outside is virus-like, but the inside has this DNA to make channel rhodopsin. Then you inject this virus in the brain region you're interested in studying, and the virus can be altered genetically to infect only one cell type and not another cell type in the brain. Then where this injection happened, the virus will inject its DNA inside the neurons, and those neurons will then read that DNA and make this light-sensitive protein. And now those neurons gain the ability to absorb blue light and create an electrical sodium current in the presence of the blue light. And after that, you implant a kind of thin fiber optic cable to deliver blue laser light to the inside of the brain, because it's dark in the brain, so there's no blue light to activate the channel rhodopsin. And then when you turn on the blue laser light source, those neurons now absorb the blue light and create an internal sodium electrical current, and the neuron is now electrically active. And then maybe something happens when you activate those cells, like maybe the mouse sleeps more or eats less or fights, or maybe nothing happens. Then you get sad and start a different project. But so now I'll go a little more into detail of how this is done. So this is a normal virus. So the normal virus is kind of a shape like this, a geometric shape made of proteins, and then inside the virus there's DNA to make more virus, because the virus has the interest of making more virus, not of studying neuroscience. And then you make an altered virus in the lab, and inside this altered virus you put the DNA to make that blue light sensitive channel from the algae. So now we have a virus that has a DNA to make that blue light sensitive protein. And after that, you get a mouse which has a brain like this, and maybe you want to study what these red cells here in this brain region does. So notice that in this brain region there's many different types of neurons. There's a yellow neuron, and a blue and a red one that might have different functions, and you want to study what these red neurons do. So in the lab you can genetically engineer the virus so that it preferentially affects the red cells rather than the other types of cells. And then you inject this altered virus there. And after you inject this altered virus there, the virus will infect preferentially the red cells and will make them produce this blue light absorbing protein from the algae, which is now shown here in green. So now you can study these green cells, which are the infected red cells that were infected by the virus. So basically the virus can be engineered to affect only one type of cell and not the other. So then when you inject this virus in the brain, it will make that one type of cell become light sensitive. So in the example we talked about before, it would be something like you can inject a virus here that would infect the red cells but not the blue ones. And then we would be able to use this laser light to activate the red cell, but not the blue one. So this virus gives us this specificity of studying one neural type, but not the other. Yes, you have a question? Mm-hmm. This size? On the right. This size? Oh, yeah, okay.
On the right picture,
Mm-hmm.
the size scale is about 50 microns,
Yes.
right? I'm curious, when you inject something,
Mm-hmm.
do you have a sensibility enough to decide, oh, now I'm injecting a virus to this red cell and not to the blue
No,
cell?
no. You don't inject the virus to the red cell. You inject the virus in this region, not one red cell, and then the virus will infect red cells that are nearby this region. so it's not that you'll infect one red cell within this 50 micrometer little space. It's more like all of these red cells that are here might be infected by the virus.
Oh, okay, thank you.
Maybe it's a silly question, but how you isolate this effect? If you put the blue light, can you guarantee that nothing happens with the brains, the cells, without these viruses, this virus? You know what I mean? Like
yeah.
first step, just putting light there and see if something happens. This
Yeah,
is
well,
like
there's
a
some
step.
other steps that I didn't explain, but first, ideally, you'd have to validate that your virus is first just infecting the cells you want and not the other ones.
Okay,
So that's an
okay.
important part. And then if that happens, then in addition, you have to use the laser light in a parameter space that doesn't cause
Damage.
off-target unintended effects. Because I mean, it is a laser light, so it can be used as a weapon and it can destroy things. So you have to use the light in some amount of strength and intensity that is not causing tissue damage or heating, etc., but is still strong enough to activate the blue light sensitive channel rhodopsin.
But these can have multiple effects. You have control on that, or
What can
the
have
blue light? The blue light can how can I say? Not perturb, but activate more than one type of cell. This
Yeah,
is possible.
that wouldn't happen if the virus only infects
If
one
you
type of cell.
guarantees that, okay.
But even if the virus infects, say, only the red cells, which is the objective, that doesn't mean the effect is only in the red cells. So the initial direct effect is only on the red cells, because only they can kind of sense the blue light. But these red cells don't only communicate with each other, they also electrically communicate with some other cells that are not shown here, and then those cells would be altered as some indirect effect.
Yeah, sure.
But that is part of what you want to study, because all the neurons in the brain do connect with some other type of neurons, and their function depends on who they connect with.
Okay, thanks.
Okay. All right. So, okay. So now we get this possibility of doing this, and then we implant this fiber optic cable to deliver the laser blue light, and then these infected green cells would then absorb the blue light and create an electrical current, and maybe that changes the behavior of the mouse somehow. And this is already something that, as you can see, uses a lot of expertise from many different fields of science. So this idea of how a photon can be absorbed by an organic molecule is something that both chemistry and physics contributed a lot to understand, and then even what is the nature of the photon is something that took a lot of expertise from physics. And this is what the rat kind of looks like after it has this fiber optic implant, and then the laser light comes from a laser to the inside of the brain and activates some cells, and then something happens. Okay, so in our lab, one behavior that we study a lot is fear-related behaviors, or fear and anxiety. And even though everyone recognizes that those are real emotional states, it's not really obvious how to study that in an animal setting. So usually what we do is that we expose the animal to some kind of dangerous stimulus, like a predator or a predator smell or something that is interpreted as dangerous, and then we study those danger-related behaviors. And those are very similar to real behaviors that those animals do in nature in the real world. So to show that, I just found a video that illustrates how that happens. So here we'll see a video of a lizard, like an iguana, and then there are some predators that want to catch the iguana. And then we'll see what it does when it's in face of this very dangerous situation, because the iguana does not want to be eaten by the predator. When the snake has not detected the iguana yet, it tries to freeze and not move so that the snake can't visually detect him. And then once the snake has already found the iguana, then he stops freezing and runs to escape. So those are two very common fear-related behaviors that animals do. And we do those things also, right? So if there's someone that's very dangerous with a gun trying to shoot people, before you're detected, you'll try to not move and hide. But then when the person has already seen you, you can't continue hiding, then you'll try to escape. So that's also what the animals do in these situations. So in the lab, we try to often study these freezing and running away escape behaviors as measures of fear in the animal. And then one region that was thought to be important for these fear behaviors is the periaqueductal gray region, which is a region in the brain stem, so very in the back of the head. So here we have a diagram of the mouse. This is the mouse brain, and this gray region here is the periaqueductal gray region that we will be talking about. And it wasn't really known which types of cells in this periaqueductal gray region control which type of behavior. So what they did here is they made a virus that selectively infected the glutamatergic cells in this region. And glutamatergic cells means neurons in this region that use glutamate as a neurotransmitter. So some cells release, say, serotonin or dopamine as a neurotransmitter, and another neurotransmitter is glutamate. And then they are studying here cells that release glutamate as a neurotransmitter in this kind of ventral or more bottom part of the periaqueductal gray. Okay, so what's happening here is there's a mouse, then the mouse has these wires that connect the laser source to the left and right periaqueductal gray ventral parts, and these cells now express the blue light absorbing channel. So in the presence of blue laser light, these cells will become electrically activated, and then maybe the behavior of the mouse will change. So here there isn't any real threat. This is just an empty box. And because there's no threat, the mouse is not scared. So it's just walking around in the box and looking at things and smelling and etc. But you'll see that when these cells become optogenetically activated, the mouse will show a different behavior. And that will be written here when it says light on. It means the blue laser light is on to activate these cells. Okay, so first the mouse is just showing normal mouse behaviors and looking at things, but when the light on happens, the mouse stops moving and it freezes as if it's trying to hide from some danger. And then the light on stops, he starts moving again and exploring and having normal mouse behaviors. So their normal behavior, just walking and exploring and looking at things. But when the light on appears, you'll see he will stop moving again. So it's as if he's trying to hide from some danger that's not there. So that was shown. And this is when you activate these cells that are on the bottom part of the periaqueductal gray. But there's also a upper dorsal part. And then we'll see in the next video that if you activate this upper part of the periaqueductal gray optogenetically with blue light, a different behavior happens. So here, they're activating glutamatergic cells also, but in this different part of the periaqueductal gray. So in this video, there's this box, and there's no real danger. It's just an empty box, and the mouse is walking around the box looking at things whenever he wants to go to different places by his own volition. But when the mouse reaches this red part of the box, the scientists are activating the blue laser, which will activate these other groups of cells, the periaqueductal gray. Then you'll see that something will happen. Okay, so mouse is here. Then whenever he reaches this red region, they'll start the stimulation. And then you see it causes a rapid escape. Like whenever the mouse reaches there, if you activate this region of the brain that causes escape, then escape happens. And this is another sort of result that was found with this optogenetic finding. Okay, so the first video was what happens if you activate the glutamatergic cells in this region. which are cells that release glutamate as a neurotransmitter. But there are also other cells in this region, the same exact region here, that use other neurotransmitters. And one of those neurotransmitters is a neurotransmitter called GABA. So we, in our lab, studied these other cells in this region that use GABA as a neurotransmitter, and then we'll see that that caused a very different effect. So these cells turned out to be involved in food-seeking behaviors, which is very different from freezing, right? So and this is a video showing that there's a mouse here, then here there's a little insect, which is a cricket. So the mice is very interested in hunting crickets, but this mouse is not hungry, so it's not really super motivated to hunt the cricket. So you'll see that during the laser off period, he has some interest in looking at the cricket, but when the laser on starts, you'll see that the behavior changes very quickly. So when the laser on starts, you see he's very interested in hunting the cricket and immediately chases the cricket and eats it. So these are cells that are right next to the cells that cause that freezing behavior in the same region, but if you activate these other cells, it causes completely different action, which is not related to freezing, which is hunting crickets. And here you can see the blue laser illuminating the head of the mouse with the blueness. So that's so this is the kind of thing that could not be found before without optogenetics, because now we can activate one cell type and not the other. Sorry? Oh, yeah, now we are continuing to study this and seeing what sorts of things it causes the mouse to want to eat. And we'll do that in the next slide. But yeah, so this was the first finding we had with these cells, which is, it seems to increase this hunting, which is one type of food source. But then we also exposed the mouse to, say, two different foods. So here's a chocolate, and here's a normal kind of mouse chow, or which is their food they eat every day in their cage. So this is a mouse that's not hungry, so because he already ate before the experiment. And then when we activate these cells, before activating, the mouse is not hungry, but when we activate it, he goes straight for the chocolate, needs more chocolate, but not more of his normal food. So this suggested that these cells cause eating more of these highly caloric foods, like chocolate, instead of their normal, well-balanced food. So here we kind of measured the amount of food eaten when we activate these cells and for different types of foods. So in the top, we have foods that are kind of healthier, like broccoli or carrot or bell pepper. And then when we activate these cells, so which is the red bar, and then you see they do eat a little more broccoli when we activate the cells compared to not activating it. But look, but the number scale, see, it's around maybe 30 milligrams of foods, something like that. But when we activate, and there's these more caloric foods around, like cheese or sausage or sugar, then they eat maybe around 100 milligrams of that food when the stimulation happens. So this shows that even in mice that are not hungry, the activation of these cells causes them to eat a lot of these caloric foods, but not necessarily of these kind of healthier vegetables. So that's one type of finding we did in our lab using optogenetics. And then another line of research we had was to study circuits that control this escape from dangerous situations. Okay, so in normal life, when there is some danger, like let's say there's a fire there, so it's very dangerous and you want to escape, you can't just run in any direction. So you can't run in the direction of the table, right? You have to make a map in your mind of where is the exit door, these are the stairs, then I'll take this particular route to escape. So you have to know which direction and how to escape when you run. And this is also what animals do in real life complicated situations. So here we'll see there's a cat, and then the cat is trying to catch a rat, which is behind here. Then we'll see how he catches it. So see, the rat came out and came back in. And the cat couldn't catch it. Then the rat will come out again, and then the rat figured out this very smart way of escaping by kind of escaping three-dimensionally in the other direction. So in real life, both animals and people are able to use the geometry of the environment to understand how to escape. But in the lab, typically escape is studied in very simple environments like an empty box. So we wanted to study how this kind of behavior worked. So first, we put the mouse in a complicated place and tried to see if stimulation of the dorsal periaqueductal cells that caused escape previously would also cause escape in a complicated situation. And that's what's shown kind of in this video here. So remember, this is how escape is typically studied in the lab. So there is this empty box here. And then when the mouse reached this place, the stimulation happened, and then he just escapes in this very simple situation. And the escape here is being caused by the neural activation, not by real danger. So we did that, but in a more complicated place. So we activated those same cells, and here's a very complicated place, lots of blocks, and then the mouse is walking around, and during the light off, he's not scared. But when the optogenetic stimulation of the periaqueductal gray happens, you'll see he wants to escape, but doesn't understand how to do it. So now he's very agitated, he's running around, he's trying to escape. But activation of these previously studied escape circuits don't make the escape in a complicated environment. Because to escape from here, the mouse has to climb this block, then this one, and then climb these orange stairs, and then jump out of the box. So it's very complicated for mouse standards. And stimulation of these previous circuits that were being studied in escape don't seem to cause escape in this more complicated situation. And this is more like real life, because we don't live in empty boxes, we live in geometrically complicated places. So we found a region in the hypothalamus that if you activate, causes escape in this more complicated situation. So here, in this video, this is the same experiment, but now we're activating a group of cells in the hypothalamus that seems to cause complex escape. So first, the mouse is walking around and looking at things as not very scared. But then when we activate these hypothalamic escape cells, it causes the mouse to very quickly understand what to do to escape, and then he escapes from the box. And this is a completely novel environment. The mouse has never seen this box, but we find that some circuits in the brain only cause this urge to escape, and the mouse runs in random directions. But we found this circuit that causes the mouse to kind of correctly escape through the geometrically most efficient route in this more complicated environment, which is more similar to what would happen in real life. And the way we noticed this happening the first time was that if you put the mouse in an empty box and then activate these hypothalamic cells, then the mouse kind of gets agitated and runs around and tries to find an exit route, but can't. And then there is no real correct way to escape here, because the box is too high, so he cannot escape. But he tries, and then he just tries to jump to escape. So here, again, the escape is being induced by the neural stimulation, not by real threats. Okay, but then this could suggest that, oh, this stimulation causes jumping. But then we got the same mouse in the same box and put a kind of route for it to escape, which is climbing this rope. And in this situation, in this situation, the optogenetic activation causes the mouse to not to jump, but to understand that if I climb this thing, then I can escape from the box. So he climbs this thing and then jumps out of the box. So that gives us the idea that these cells might be controlling this more complex kind of escape, which we showed in the previous video. Okay, so the last kind of finding I want to show with optogenetics was done by a different lab from Caltech, where Richard Feynman worked. And a neuroscientist in Caltech found a different nucleus in the brain that if you activate, causes very violent attack. So here we have a mouse, and here we have a lab glove, which is not something the mouse usually attacks. So the normal behavior of the mouse is to just investigate this object and see what's happening. But if you activate this nucleus in the brain, it causes this violent attack towards this glove, which is something the mouse wouldn't normally do. And then if you put a different mouse in there, he also attacks the other mouse. So they found some nucleus in the brain that causes attack. And something interesting about this, that these are not just things that happen in a mouse in a lab, but to the extent we can tell, these sorts of effects seem to happen in people as well. So for example, when I showed that video of optogenetic stimulation of the periaqueductal gray causing freezing and escape in the mouse, it suggests that the mouse is scared of something. But we can't really ask the mouse if it's scared. But in a neurologist's office, this was somewhat simulated in a human. So in a human patient, if you electrically activate that same region in the brain, the person has a full-blown panic attack. So even though the person was previously happy, if you activate that region in the brain that causes escape in the mouse, it causes panic in the human. So the human says things like they might die at any moment, it's very unpleasant, there's something chasing me, I think I'll die at any moment.
Sorry
so
for the
they
curiosity.
Get scared.
How you activate that in humans?
You activate that in a neurologist's office, kind of surgical suite, with the electrical electrode just kind of...
So it was done during a surgery or...
Yes.
I mean, I see.
Yeah, during a surgery.
I see.
So but they didn't do it to cause panic attack in the patient. They did it...
Oh, I can
the
imagine,
reason they
yeah.
did it is... But it's an interesting reason why they did it, too. So they did it because that same region was also known to induce analgesia. And then if you can induce analgesia, that might be good to treat chronic pain problems. And that same region then that creates this panic also creates analgesia.
This is something that is coming out recurrently. It looks that, I mean, very opposite things stay in the same place.
Yes.
Is there any reason for that? Or
Well, in this case, the reason is pretty understandable, which is that you want to have analgesia when you are panicking.
I see.
Right, because let's say there is something very dangerous, the people are shooting guns and it's very dangerous, so you want to escape. And at that moment, the priority should be to escape. So if you're escaping, you shouldn't go, oh, my knee hurts from arthritis and this thing itches. And so you want to just be focused on the escape. So in situations of danger, people do have very intense analgesia. So in that case, it makes sense that those two things happen together. So that's why they stimulate this region, is to study this analgesic effect, which does happen. The person does get also analgesia. But they also become terrified, so it doesn't work as a clinical treatment. But to the extent we can tell, these same types of brain regions do at least qualitatively similar things in people. So in conclusion, what we wanted to talk about is that you can use this method called optogenetics that was developed with the expertise from many different subjects, including biophysics and chemistry and physics and microbiology, to develop a method that allows you to selectively activate only one particular neuron in the brain and see what that population of neuron does in a behavior. And this has already identified many circuits in the brain that control all sorts of different behaviors, like addiction to drugs, or depression, or eating, or sleeping, essentially every behavior you can think of. And in our lab in particular, we have used these tools to identify regions and circuits in the brain that control escape from threats in more complex geometrical environments. And also we found circuits that can induce eating even in the absence of hunger. So I'd like to thank everyone in the lab that did all these experiments, and also our funding sources that paid for the people in the lab so they don't become all homeless. So happy to take any questions that you might have.
Okay, thank you very much for this very interesting talk. questions are already proliferating, so let's start.
So the virus that targets a specific cell was a very important tool
Yes.
for doing what you want to do, but could you make a comment on like how easy it is to build such virus? It wasn't clear to me if there's something you can just go buy off a shelf or if it's something that takes hours of
Yeah, that's a
time.
good question. So I didn't go into the technical detail of how that happens, but it is easy because we are not exactly making a virus that infects only this neuron and not that neuron. So that was a kind of simplified explanation. So what's happening is you can purchase from companies transgenic mice that already produce a certain specialized protein only in one type of neuron. So you can buy a mouse from a company, for example, that has a special artificial protein in all dopamine-releasing cells. And then you can make a virus that will infect only cells that have that artificial specific protein. So you can use that same virus to infect this other type of neuron if you purchase a different mouse that has that artificial protein all in that other type of neuron. So you don't need 5,000 different types of virus that are independently engineered to infect 5,000 different types of cells. Rather, people have made many thousands of lines of mice that can be infected by that one type of virus. And then if you get, say, yes. So it's more that people made many types of mice more than many types of virus.
Yeah, very nice talk, thank you. I'm thinking about, let's consider the experiment in which you had the rat in an empty box and then the one that you put some things and the rat climbed. I mean, you are assuming that this different type of behavior is due to the type of interaction that you do on the cells of the rat. My question is, why not to assume that the rat may have some intelligence?
The rat might have some what?
Some intelligence.
Well,
Some level of intelligence.
But we're not assuming that he doesn't have intelligence.
Sorry?
But we didn't assume the rat does not have intelligence.
No, but I mean, is the fact that the rat detected that there was some way to climb out of the box, was it due to the fact that you were putting the light on the cells, or was it due to the fact that the rat has some intelligence?
Oh no, it's both. It's both things happen. Yeah, because when there is no danger, the rat has no motivation to escape, so he wouldn't even try to escape. But definitely a real rat is able to understand how to escape correctly. But the issue is when people were studying these escape circuits in the lab, they were studying circuits that only caused this very disorganized escape, that caused the rat to just run and bump into a wall and then run here. Then the rat wasn't able to, the previous brain regions that were being studied didn't cause this more natural type of escape, which is what the rat normally does. And then we found a very obscure nucleus in the brain that seems to induce this real behavior that does exist because the rat is intelligent and can find what's the way to escape.
Okay.
Yeah, thank you for the nice talk. I had a really naive questions about if you think about, for example, the changes in the amplitude of the blue light. Like, for example, if the amplitude of the light is really low, then the rat is not afraid, but then if you start increasing it, it will be more afraid or less afraid.
Yes, we tried that with this escape circuit, because sometimes when you stimulate the blue light using different parameters, sort of qualitatively different behaviors could happen. But in our case, we saw the same behavior, which is escape, happens at different amounts of intensity. Okay, so let's say rat is here in this cage and you're doing nothing. Then the rat's very happy in the cage, just walks around and looks at things and is okay. Okay, so then we saw that if you stimulate a little, it causes the rat to become slightly more exploratory. Like he might do this against the wall. Like he does more of, I want to see, okay, then he goes back. Then we saw that if you stimulate slightly more, then it increases the propensity of the rat to escape. So then he goes up here and maybe he climbs on the wall. Then he's on the wall looking at what's, and then if you activate more than that, then cause the rat to jump from the floor to the outside. So it seems to not cause a different behavior in this case, but less of the same behavior. But in other circuits that are studied by other people, it was sometimes found that different parameters of light stimulation can cause qualitatively different actions. So it was found, for example, that there is a group of cells in the hypothalamus that if you stimulate in one kind of weaker way, it causes the male mouse to display sexual behaviors to the female mouse more often. But then those same cells, if activated more, cause the male mouse to attack the female mouse, which is a different behavior. It's not more of the mating behavior. So yeah, in different circuits, you could have very different results with different amounts of light stimulation. So it's always good to try to see what your circuit is doing at different stimulation parameters.
So I have a question about how do you determine both the precise cells that are involved in a specific behavior and the specific function that is involved. So what I mean is the following. You just said that there are, in principle, the specific neurons are connected to many other neurons. Mm-hmm. So in principle, there could be secondary effects. Mm-hmm. So there's two questions that come to my mind. First, how do you know that the specific behavior you see is related to the neurons that you stimulate and not to neurons that are secondary connected? Mm-hmm. And the second, how do you know that the specific behavior you see is not a secondary function? Let's say, for instance, you see, you increase, the blue light makes things look more clear, and so maybe finds quickly the way out, rather than because he is more motivated to find a way out, right? So how do you...
Yeah, very good question. So the first question is, say we're stimulating this neuron of type 1, and then how do we know the behavior is caused by that and not by neurons connected to neurons of type 1? And then the answer is, those are not really two different things, because the function of the neuron 1 normally is to distribute its activity to whoever it's connected to, right? So for example, the reason in the car that if you turn the key, the engine starts, is because the real function of the key is to cause this other thing in the car to also work. So the reason any neuron causes any behavior is because their activity is propagated beyond themselves to some other cells.
Like in principle, if this is true, you could activate directly the second neuron.
Yes, yeah, that would happen too. But not all the neurons are causing all the behaviors. So, but yes, if this neuron is causing a behavior by acting on this one, then yes, stimulating this one also often causes a similar action, yes. But not necessarily, because one neuron can also electrically connect to this and that one. And then you might need both of these to be active to cause the behavior. And then if you activate only this one, maybe this one doesn't activate that third one. And then maybe only a sub-part of the behavior would happen. Yes, so whenever you see some behavioral effect, you want to be sure that it's happening for the reason you think and not some other different underlying more basic action. So for example, in the eating result that we found, that if we activate these cells, it causes more eating, okay? So there could be many reasons why the mouse is eating more. So one reason could be that, oh, perhaps he's less scared, so he's more willing to go out, and then that's why he's eating more. So that's a very possible reason. And then we know it's not that reason because when we tested the effect of these cells on just more pure fear behaviors, so there's no food, just threat that causes fear, then when we activate those cells, it doesn't cause less fear behavior. So we know the eating is not being caused by less anxiety, which is a completely different action. But okay, so that gets rid of one confound. Then, but could be another confound. Could or not confound, but there could be another reason. So another reason could be it's eating more because it's causing more hunger. And then we tested that by seeing if the stimulation is positively or negatively motivating. Okay, so hunger does cause eating, but hunger is unpleasant and mice don't like to be hungry. So if the stimulation is causing a lot of hunger, then the mouse sort of wouldn't like the stimulation. And then we did do a experiment for that I didn't talk about, but I could mention now, which is shown here. So here we have a box that has two chambers, and then the mouse can freely go from left chamber to right chamber. And then whenever he enters the right chamber, we then turned on the stimulation. And then the colors here represent the amount of time he spent in each pixel. So in the control situation, he sort of spends the same amount of time in the left and right part of the box. But here what happens is we now turn on these cells optogenetically whenever the mouse goes into the right part of the box. So if going in the right part of the box is very unpleasant and bad for some reason for the mouse, then he will avoid going to the right part of the box. But instead, he seems to spend more time on the right side of the box. And there's no food here, it's just empty box. So the fact that he spends more time where there is stimulation indicates that this is not causing some intense hunger. So that's not the reason why he's eating more. So it seems so, because it's not hunger, the other main reason the mouse might eat is for kind of pleasurable pursuit of tasty rewards. So that's the reason we are attributing it to now. But you're absolutely right that whenever you see a behavior, you want to see if there is not some other underlying action that could explain it. And then you try to get rid of the more obvious confounds, but there's some infinite number of things that could be explaining it. So another thing we try to do is also try to see if the same qualitatively similar type of result happens in a very different setting, like in a completely different box, completely different food, completely different set of movements that is needed to get the food. So we try to do also that, try to show that the effect happens in a different situation.
Okay, I was also curious about the same question Dario asked about these cells with opposite functions living really close together inside the brain. And we constantly hear about many functions in the brain working against each other, like in a seesaw way, like hunger suppresses disgust suppresses. Is that why they sometimes are close together? Is that linked at all? Or?
Yeah, because the easiest way to suppress this behavior is by being near to the cell that creates that behavior. And this competition model, I think, is very accurate because we don't, in some sense, you think about it, it's not, we don't really have like a unitary mind. We sort of have different modules in the brain that are somewhat loosely connected, and they have their own internal motivations and things they want to do. and they compete with each other on influence on the behavior. And you can notice that on yourself whenever you have some conflict of, oh, I should be doing this, oh, but I should be doing that. Those are different parts of your brain that want to influence the behavior in different ways. And this is probably one of the biggest problems that people have, which is, why can't people do the thing that they want to do? It's because they also sort of don't want to do it. Right, so for example, when it's the beginning of the year in January, people make these resolutions that I'll be super healthy and I'll go to the gym and I'll eat a lot of carrots and I'll do a lot of exercise, and they want to do it. But then in February, no one is doing that because they also don't want to be doing it. They want to eat ice cream and sleep. And then there are these different motivations that compete with each other.
More questions? Okay, if not, I can ask maybe two questions.
Yeah.
One is, I mean, how you said that some group before you targeted this area in which the mouse was escaping randomly, and then you found that hidden in the hypothalamus there was.
Yeah.
How you came, more generally, how you decide to target one cell with one thing instead of the other? What is the, I mean, historically, for example, this poor guy got, a damage. So is there a rationale to decide to target one area instead of the other, or is more like trial and error?
It's a little of both. So,
for example, in your specific case, how you decided to go to check the hypothalamus to maybe find a better way to escape, if you can say.
Okay, in my case, it was accident.
Okay.
So basically what happened was this. So I wanted to study circuits that caused escape. So
Okay.
That was true, because escape was less studied than circuits that controlled some other behaviors. So that seemed like a good area of research. And then I read a paper that there is a particular nucleus in the hypothalamus that there was just one paper showing that if you activate it, it caused escape. So I thought, there's only one paper on this, so I could explore this more. And then I tried to replicate that experiment. And then I saw that, oh, it does cause more escape. So this is good.
I see.
So it looked like it worked. And then the original paper is just in this empty box, just running around escape. So I thought, oh, what would happen if we put it in a slightly more complicated box? Like if we add a rope, would it then do this?
I see.
And then I saw that you could put that mouse in any arbitrarily complicated place, and it very quickly figured out how to escape from this complicated place. So that seemed a completely new finding that the other paper didn't have. Okay, so then that's where the accident part comes in, which is, so the hypothalamus is not a one thing, but it's rather a collection of
Exactly.
many different nuclei that are very closely together. So then when I made brain slices to see where was the injection of the virus in those mice, it wasn't in the part of the hypothalamus that I was trying to target. It was sort of in a slightly different region that was very understudied and people didn't really know what it did. So it was a different area of that bigger hypothalamus. And then that part was by accident. Or the eating circuit was also by accident, because, see, in the eating circuit, what was known was that if you activate these glutamatergic cells here, it causes freezing. So this was known by other experiments from other people. Okay, so the glutamatergic cells are cells that release this glutamate as a neurotransmitter, and this is a neurotransmitter that electrically excites cells, which means cells that bind glutamate, they have positive charges going in them like sodium, which makes them more active. But there are also neurotransmitters that are inhibitory. So if you bind those neurotransmitters, kind of maybe negative charges go inside the cell and make them less electrically active. So the main glutamate is an excitatory transmitter, but GABA is an inhibitory transmitter. So we thought that, oh, if you activate these glutamatergic cells here, it causes freezing, which is what other people said. So we thought maybe if you activate the inhibitory GABA-releasing cells here, it should cause less freezing, less escape, less fear, etc. So that was our idea. But then we saw that if you activated those inhibitory cells in this region, it didn't cause less freezing, less escape, less fear. So then we were very sad because nothing happened. But we also observed that it's not that nothing happened. So we didn't see less freezing, but it did seem like if you pay attention, the mouse becomes more exploratory and looks at things more. And if you put an object in there, he sort of explores it with his mouth more. He's like, let's see what this is. He does more of that. And then by exploring that more, we found that it causes this eating type of behavior.
I see.
So that was sort of accidental.
Mm-hmm. And the other question is, I mean, it's clear that, let's say, I guess the long-term goal is then to do some targeted therapy for whatever. I mean, you realize more specifically what is implied in what, and so hopefully there will be more, let's say, therapies for various diseases. Can you estimate the time span from, let's say, a discovery like this and a possible therapy?
It depends. Okay, so this cannot directly be done in a human because
I know.
because you can't put people don't want to have lasers
Sure.
in their brain and they don't like it.
Sure.
But a lot of these discoveries are likely applicable in humans. As far as we can tell, the humans have the same circuits. So probably activation of these cells does cause a panic attack in a human. So probably, say, inhibition of the cells we studied would cause less food seeking in a human. So the issue is that this viral targeting issue doesn't really work in a human at this moment. But there is a lot of effort to try to do it. And then this would solve not problems in neuroscience, but would solve lots of problems in medicine in general. So there's a lot of research trying to find viruses that can be used to deliver copies of certain genes to certain organs, right? So for example, there are a lot of people that have one enzyme that doesn't work in the liver, and then because of that, they have a big metabolic problem. And then if you could deliver through a virus one copy of the correct gene, then that problem would be solved. So people really want to solve this. And then so I believe that would be solved, this problem of how to use viruses to infect specific cells in human patients. And then when that's applied to the brain, the way it would likely work is not through lasers, but it would be through some kind of oral drug, which is what humans can take more readily. So the idea would be you can make a virus that infects, say, these glutamatergic cells here, and then put some kind of drug receptor on that cell. And then when you eat the drug would go through the bloodstream and inhibit that cell. So we can do that. We can make molecules that inhibit neurons. So that part is solved. How to get the virus in the correct cell type, the human is the part that's not solved. So once that is solved, which probably will, I think, in, say, less than 20 years or 15 years, because there's like hundreds of billions of dollars of money that would be made if this is solved. Yeah. So, and people have solved more complicated, stranger things than this, you know, because the idea here, it doesn't even sound that weird. You engineer a virus that infects these cells in the body. Like that, it doesn't even sound that far-fetched. But there's a lot of, long-term safety concerns that you need to look into, because the mouse only lives like 1 year, and then in 1 year maybe it's not enough time for the virus to cause a problem. But the human can live 80 years, so you don't want to put a virus that 10 years later causes more cancer. So we do have to solve those problems. But once it does, there's like thousands of these findings from the brain that could be tested in a human to see which one would work and not. So on one hand, it seems like the knowledge about the brain didn't increase that much from 30 years ago because the treatments are not that much better. But on the other hand, we do know a lot more about what circuits control more cocaine addiction or less, or more sleeping or less.
So you're preparing the ground.
So, and a lot of those things probably work in the humans too. It's just we don't know how to target those cells. But I think after some decent type of viral therapy for gene delivery is discovered, that would be applicable here.
Okay, thank you very much.
Yes, thank you.